Good morning, everyone. Here is Group 2, and in today's presentation, we are going to give you some insight of the fluorinated flame retardants. Those retardants are also known as per- and polyfluorinated alkyl substance, aka PFAS, which is a large and complex group of synthetic chemicals. They are widely used for their property of high thermal, chemical, and biological stability. For instance, daily life application of PFAS includes making clothes that are resistant to stains and producing firefighting foam that is durable to high temperature. PFAS molecule consists of a chain of linked fluorine and carbon atoms, which is one of the strongest chemical chains, and thus making the molecule long-lasting in the environment and causing public health concerns. In this presentation, our group will firstly introduce the uptake pathway of PFAS in the plant, and then discuss the PFAS potential in the ecosystem, followed with remediation technologies from different aspects to mitigate its contamination.
Hi, I'm Tianqi. As Johnny said, PFAS is widely used in various industrial products and household items due to excellent chemical and thermal stability. Therefore, I would like to introduce the sources of PFAS in plants and the uptake pathway in plants. Actually, there are several sources contributing to the accumulation of PFAS in plants. First, emissions from industrial manufacture can contaminate the surrounding soil, water, air, and consequently plants. This is the main sources of PFAS contamination. Second, field irrigation with PFAS-contaminated water represents an important pathway responsible for PFAS accumulation in plants. The massive production and long-term use of PFAS has led to their ubiquitous presence in freshwater bodies, such as lakes, rivers, and groundwater. Third, land application of biosol is a common practice to improve the soil quality and is another major pathway of introduce PFAS into agricultural fields. Moreover, litter from landfill sites has been identified as a point source, contributing to the dissemination of PFAS in the environment. All above are major source of PFAS, and in fact, this has become an environmental issue that has to be taken seriously. By reading the articles, I found that it seems to be a global pollution problem already. For example, in China, USA, Spain, and Uganda, high concentration of PFAS has been found in various environmental media, in soil, surface water, groundwater, and air. Therefore, studying in the source of PFAS is important to the uptake pathway in plant. Root uptake from soil and water is considered the primary pathway of PFAS to enter plants. PFAS in soils can migrate towards plant roots, driven primarily by the water potential gradient created by plants' transpiration and PFAS concentration gradient. Well, the uptake mechanism of PFAS into plants roots remains largely unexplored. Through some studies has initiated mechanistic research. Lesser amounts of vapor phase and particular bonds PFAS can also be solved by the above ground portion of plants, such as leaves and bark. The amino contribution of foliar uptake to plants accumulation of PFAS could be due to the short contact time between vapor phase and particular bonds PFAS and plant above ground surface. The resistance of wax cuticle in foliar and the relative low concentration of airborne PFAS. Okay, moving on to the next part.
We'll now move on to the PFAS distribution within the plants. According to the previous studies, PFOA is one of the most well-studied PFAS because it has been manufactured the longest. The result shows that for agricultural vegetables, more than 80% of the PFOA contaminants accumulate in the leaves. For arbors and shrubs, the total concentration of 26 different PFAS contaminants accumulate more in leaves and twigs than in trunks and roots. A similar phenomenon also applies to conifers. The contaminants distribution can be explained by two different influential factors. From the plant's aspect, the existence of the preferential accumulation of PFAS in vegetative structures of plants could be partly due to the enrichment from the greater transpiration stream. The plant structures which receive a larger amount of water to the sides could accumulate more PFAS. From the PFAS contaminant aspect, according to the existing research, long-chain PFAS more readily accumulate in roots, whereas short-chain PFAS has a high capacity to be translocated from roots to shoots. One parameter that links to the influential factor is the translocation factor TF, which is calculated as the ratio of PFAS concentration in shoots to that in roots. In the previous studies, when the type of plants being examined are edible crops, such as radish, celery, and tomato. According to the graph, the long-chain PFAS, such as PFOS, has low TF value. These contaminants with low TF values tend to accumulate in the root part of the plant rather than in the leaves. Influential factors that affect PFAS accumulation in plants are as follows. PFAS with different chemical functional group has different translocation and accumulation effects in different plant species. For instance, PFOS would accumulate more in the roots and PFOA more in the leaves due to functional group issues associated with binding potential and translocation. Apart from that, short-chain PFAS generally demonstrate a higher accumulation potential in plants compared to long-chain homologs. Besides, in general, increasing salinity reduces plant transpiration, which might consequently diminish plant uptake of contaminants. Temperature can promote plant physiological activities and biochemical process, such as increasing transpiration and metabolism rates, which in turn influence plant uptake of PFAS contaminants.
Hi. After my partners explain about PAFs, I believe you have a basic understanding. My part is about PAFs potential in ecosystem, and it is divided into 3 parts. What is PFAS potential? Factors of PAFs potential and ecological risk assessment about PAFs in aquatic and terrestrial systems. Now let's move to the first part. Numerous studies show that PAFs are ubiquitous in aquatic and terrestrial environments. It is detected not in just near the source of pollution, but also in nearby ecosystems like a river or a lake. Furthermore, it has been detected around the globe in like drinking water, in human body, and also in non-human biota tissue. Thus, it is necessary to know the trophic transport and biomagnification potential, which is PAF's potential in this topic, and it will help the ecological risk assessment and pollution abatement. To analyze PAF's potential in ecosystem, I choose biological specificity as qualitative analysis and hydrophobicity as quantitative analysis as influential factors. For biological specificity, the results shows that there is an increase of PAFs from down the food chain to up the food chain. However, different food chain will show a different trend. Hydrophobicity studies gives that there is no relation between BMFS and TMFS with the potential in aquatic system. There are also some other factors like creature size, quality. However, with limited sample study and high uncertainty, it is unpractical to do analysis about it. After analyze these factors, there are some further study which can be improved. For example, consider the influence of PAFs enrichment in biological system, and if human intervention will decrease PAFs potential in ecosystem. Meanwhile, uncertainty analysis about those factors is also necessary. The residence shows a natural accumulation with the process of food chain in both aquatic and terrestrial system. However, different food chain and biotic community shows that different PAFs potential due to several factors: hydrophobicity, biological specificity, And so distributed data is needed for a comprehensive understanding of PEF's potential in ecosystem. Now let's move to the next part.
Here are the remediation technologies, and there are three parts. First one is physical method, and second one is chemical method, and the last one is bioremediation. And for physical method, I classify them as some for water and some for soil. Due to time limits, I can only describe some of this method. First one is activity carbon, which includes powder activity carbon and granular activity carbon. The high absorption capacity of active carbon is the result of its internal pore characteristics. Cost saving is an advantage, and it's effective for the removal of long chain PFA, but it's not effective for short chain PFA and precursors. And second one is nanomaterials, which are mainly based on carbon nanotubes. Compared with other absorbents, CNT has fast absorption equilibrium and higher absorption capacity, but the cost of manufacture is high. And next one, the ion exchange resins. First is the principle of IXR, and it's a well-established technology, and it's more effective at removing long chain PFAS. The disadvantages of IXR are that secondary waste is generated, and its performance is strongly influenced by the presence of contaminants. And for the next one, foam fractionation relies on bubbles to absorb PFAS. And for ozone fractionation, turning the rejected air bubbles into ozone bubbles, this technique can be used to concentrate or separate components from a mixture based on selective absorption. However, the generation of foam waste requires further treatment. Next one is filtration, which is mainly about membrane technology. The two main techniques are nanofiltration and reverse osmosis, which both operate at no pressure. Membrane technology is a proven treatment technology, but it has relatively high capital and operational costs. For used membranes, it requires further treatment and disposal. Next one is soil washing. The contaminated soil will be cleaned with water, and the recovered water will be treated. It's easy to operate, but it needs large investment and large amount of wastewater treatment requires. And next one is ball milling. In this method, a cylindrical vessel containing steel balls and the material is rotated, and the impact of the balls with the soil particles reduces the size of the particles and reacts on the surface of the ball mill. It has low installation and running costs, but field application are limited. And next one, absorption and stabilization. This method also uses absorbance. The difference is that the PFAS needs to be fixed in place, and this will result in PFAS staying on site, and the long-term efficiency and stability are untested. And the last one, thermal treatment. The soil is heated to 500 to 600 degrees to evaporate PFAS due to the high temperature required. The method is expensive and requires a high initial investment. However, the capture of waste gases is challenging.
Hello, I'm Xuankai Wang. In this part, I will introduce the technologies of remediation using chemical method. There are two different media needed to be remediated. The first is for water. There are three types of methods: electrochemical method, sonic chemical method, and advanced oxidation processes, which will be shown in details in the following slides. The other media is soil. Chemical oxidation is the main method, but it is in the experimental process. Therefore, I will not discuss this one. For water, it is mainly used electrochemical method. This method uses a sequence of chain reaction, which can be summarized by the given chemical equation. This method has two advantages and three disadvantages. Its degradation efficiency is improved and can solve a wide range of micropollutants. On the contrary, it is less effective for shorter chain PFAS. What's more, electrode corrosion and toxic byproducts are not able to be ignored. The second chemical method for water is sonochemical. Its principle is violent bubble collapse, which producing oxidizing radicals. Hotspot theory is introduced to discuss sonochemical reaction. It explains violent collapse of generated cavitation bubbles with high temperature about 5,000 K and high pressure up to 1,000 atmosphere. The process is shown on the left. The sonochemical reactions are in three zones: core of cavitation, bubbles gas phase, bubble liquid interface, and bulk solution. The cost while using this method comes from high temperature and pressure. Advanced oxidation processes is the third method. It produces stronger and non-selective hydroxide radicals. Using this method, pollutant is degraded into non-toxic smaller molecules or carbon dioxide and water. The main problem is the demand of energy.
Bioremediation, which is defined as a process of using a biological agent such as microorganisms, plants, bacteria, or fungi to destroy or immobilize contaminants. Unlike chemical and physical treatments, which are often energy-intense and expensive, bioremediation typically has low initial costs and minimal equipment. It represents a simple, environmentally safe, and cost-effective approach to treat PFAS-contaminated environments. Based on biological agents, the bioremediation could be divided into three categories, which are plant remediation, bacteria remediation, and fungi remediation. Plants have been reported to remove a wide range of PFAS. The process was predominantly achieved by bioconcentration. Plant roots can uptake PFAS molecules through both passive and active mechanisms, when they are growing in the soil or aquatic system. In this way, the PFAS could be translocated and accumulated within plant tissues. Study also shows that some interaction between plant and microbe could help to improve the removal of PFAS. For instance, in some wetland system, microbe can help to degrade the PFAS content in the plant tissue, leading to their mineralization. On the other hand, bacteria species have been reported to degrade PFAS under either aerobic or anaerobic condition. Recent research by Shaw et 2019 found a species that could degrade one certain type of PFAS chemical at a very high efficiency within 1 week. Although various hypotheses for biodegradation are unclear, experimental results indicate the possibility of rapid bioremediation carried by bacteria. As for the fungi, there is only a few studies have considered the overall role and capacity of fungi remediation, and even much less for the PFAS degradation. The fungi remain a prospective candidate for remediation, as it already plays a vital role in the ecosystem by breaking down many complex compounds in biogeochemical circles. Just as other remediation technologies, there are some certain challenges and limitations for bioremediation. Firstly, it does not provide complete degradation. Therefore, the ideal application for it is to combine with either chemical or physical process to reach the maximum efficiency. Also, more detailed and accurate experiments need to be done to fully understand the PFAS biodegradation pathways. At the current stage, many pathways are being proposed and need to be identified before put into practical use. Besides, the bioremediation could be significantly affected by many factors, such as metabolic and nutrient availability, matrix pH, and temperature.
In a nutshell, our group investigates a variety of aspects related to PFAS, exposure, and remediation. From the exposure aspect, plant uptake is influenced by physiochemical properties of compounds and plant physiology. Biological specificity and hydrophobicity analysis as factors that influence the PFAS potential in the ecosystem shows an increase from down to up throughout the food chain. To decrease the detrimental effects of the PFAS on the environment, different remediation technologies are introduced. From the remediation aspect, the physical method is cost-effective. However, it may produce toxic sludge. The main approach for chemical method is to convert PFAS into harmless substance by oxidation. Lastly, for the biological method, phytoremediation is sustainable, but its practical applications are limited to factors such as time and toxic effects on plants. In conclusion, our studies tend to provide a comprehensive summary of the current key findings related to PFAS exposure and remediation technologies. The reference for the presentations are as follows. Thank you for listening.